this chapter, chapter 7, we're going to look at cell membrane structure and function. One of the key features of cell membranes is that they have selective permeability. Some substances can pass while others cannot. And this is due to their structure. Cell membrane structure is going to be amphibolic in that it has both a hydrophilic and hydrophobic region. So the hydrophilic region is going to like water. The hydrophobic region is going to dislike water or repel water. And the reason for this is because of the phospholipid bilayers in the cell membranes. So they're going to have the phosphate heads, which are going to be hydrophilic, and your fatty acid tails that are going to point towards the center are going to be hydrophobic. So the membrane is a fluid structure in what we call a fluid mosaic model. You've got various proteins that are going to be embedded in the bilayer, and those proteins are going to move around slower, but those phospholipids are going to move around quickly in that membrane. They will be able to switch locations. So over time, there have been a couple different membrane models. So in 1950, membranes for red blood cells were analyzed and found to have lipids and proteins in them. In the 1960s, we had the Dawson-Daniel sandwich theory that said we had a phospholipid bilayer, and between the two layers of that was found between two layers of protein, and that was widely accepted. In 1972, Singer and Nicholson proposed the membrane proteins were actually dispersed and individually inserted into the phospholipid bilayer, so you would have these hydrophilic regions protruding. So membranes are fluid. Your adjacent phospholipids can switch positions rapidly, 10 to the 7th times per second. Proteins are going to be a little bit slower because they are larger. So many of the proteins drift in a highly directed manner, while others are going to be attached to the cytoskeleton. So it's not random how they move around. And membranes are going to remain fluid until the temperature drops and it solidifies. So this is one of the reasons why frostbite is so dangerous for humans. When the temperature drops down, our membranes are not fluid anymore and they're no longer functioning the way they should. Cholesterol is important. It's going to make the membranes less fluid at body temperature of humans by restraining the movement. It's also going to hinder packing at low temperatures that are required to solidify. So it acts as a temperature buffer and allows us to survive in a little bit wider temperature range. When we look at the membrane proteins and their functions, so far over 50 kinds of proteins have been identified on the plasma membranes of red blood cells alone. Your integral proteins, these are going to penetrate the hydrophobic core of the lipid bilayer. Your transmembrane proteins are going to be integral proteins that actually span the entire membrane. And then we will have some that are not embedded at all, but more of appendages bound to the surface, and they're going to be exposed to parts of the integral proteins. The integrins are integral proteins that are going to attach to fibers of the extracellular matrix. So there's 6 major functions that are performed by proteins of the plasma membrane. They're going to transport things. They can be used in enzymatic activity, provide a way for signal transduction, cell-to-cell recognition, joining cells together, and attaching the cytoskeleton and extracellular matrix. So when we look at cell-to-cell recognition, we have the glycolipids that are going to be short membrane carbohydrates covalently bonded to lipids, and your glycoproteins that are going to be lipids covalently bonded to the proteins. They have a great deal of variation in the carbohydrates on the extracellular side, and these can function as markers. You have markers that will identify what type of cell. You can have markers that will even identify your cells as an individual, unique from any other human. So the membranes do have a sidedness to them and a directional orientation. When they're synthesized, you're going to have membrane proteins and lipids synthesized in the endoplasmic reticulum. And then they will be converted into glycoproteins. Those glycoproteins are modified in the Golgi apparatus and become glycolipids. The transmembrane proteins, membrane glycolipids, and secretory proteins are transported to vesicles in the plasma membrane. The vesicles will fuse with the membrane, release the secretory proteins, and then the glycoproteins and glycolipids will end up being positioned on the outside. So the structure of the membrane is going to result in it having this selective permeability. So when we look at the lipid bilayer, you've got these nonpolar hydrocarbons. Hydrocarbons are just the hydrogen and carbon. Carbon dioxide and oxygen are hydrophobic molecules. They're going to be able to cross easily. This is a good thing for us, as it allows us to have our gas exchange and breathing move quickly and easily across cell membranes. The hydrophobic core is going to impede the passage of ions and polar molecules. They don't want to have to go through this middle portion here. So we can see that there is a sidedness to the membranes when we look at this image. Your cytoplasmic side is going to have the C terminus, the N terminus on the extracellular side. Our transport proteins, these are going to be permeable to specific ions and polar molecules. This is going to allow some of the hydrophilic substances to avoid contact with the lipid bilayer. Channel proteins, they have a hydrophilic channel. This will allow certain molecules or ions to pass. So in this picture here, we can see we've got these transport proteins that are going to allow things to cross the membrane. Aquaporins are a specific type of channel protein that are going to allow the passage of water molecules. Your carrier proteins, these are going to hold on to a chemical passenger and change shape to shuttle them across. This is specific for substances that are translocated. So here, this one allows you in this picture just to go through the rest of it while we're here. This one is showing a membrane protein acting as an enzyme for a reaction to take place. This one showing it acting involved in signal transduction. You have the signal molecule come in and the protein is a receptor in the membrane and passes the signal on. For cell-to-cell recognition, you have this glycoprotein attached that another cell can recognize, joining cells together. And here where it's attaching to parts of the cytoskeleton and the extracellular matrix. So when things move across a membrane, you can have different types of transport be involved. Passive transport is going to involve the diffusion of a substance across a membrane, and it's a passive process, so no energy is required. Diffusion is the movement of molecules so that they spread evenly into the space available. They move down their own concentration gradient, so they'll move from an area of higher concentration to lower concentration. The concentration gradient is just that region along which the density of a chemical decreases. So it's going to diffuse down its own concentration gradient. So we can see here when we're looking at just one substance, you've got these molecules of dye, the semi-permeable membrane. They're going to want to diffuse across so they can be in equal concentrations on both sides. Once you reach that equilibrium, it doesn't mean that they're no longer moving, it just means they move at the same rate in both directions, so you have no net diffusion. If you're looking at two solutes, here you've got the yellow solute on the left, the purple solute on the right. They want to diffuse down their own concentration gradients. So the yellow wants to move to the right, the purple wants to move to the left. Again, they'll both move until they reach equilibrium. Then they continue moving, but they move at the same rate, so there is no net change. So when we look at the effects of osmosis on water balance, water is going to diffuse across a membrane from a region of lower solute concentration to that of higher solute concentration. So in this example here, we've got a selective permeable membrane, but we have these sugar molecules that are too large to cross through the membrane. So what's going to happen is you're going to have osmosis where you'll have diffusion of the water move across this membrane. So here on the right, you can see that there's a higher sugar concentration than on the left. What's going to happen is the water from the left is going to move over to the right to dilute that sugar. So when we look at cells without cell walls, their water balance is going to be really important because they cannot tolerate excessive loss or gain. Tonicity is just the ability of a solution to cause a cell to gain or lose water. If something's isotonic, there's no net movement of water across the membrane. It's going to go in the same rate both directions. If you have a hypertonic solution, the cell will lose water to its environment and shrivel up and die. It's going to pull the water out. A hypotonic solution, water is going to enter the cell faster than it leaves. The cell is going to swell and burst. So osmoregulation is just this control of water balance. So when we look at this example here, we've got an animal cell, which would be a red blood cell. You take the normal cell and you put it in an isotonic solution. Water is going to move in and out at the normal rate, equal in both directions. The cell will stay happy. Here, when you put it in a hypotonic solution, water is going to go into the cell, cause it to swell and burst. Over here on this side, we have it in a hypertonic solution. It's going to pull water out of the cell and cause it to shrivel and die. So we actually use principles of this in preserving food. When you make jams or jellies, one of the reasons why you add a lot of sugar to them is to throw off the osmotic balance of microorganisms that would attempt to grow in there. So sometimes people will try and attempt to make them with less sugar, and what they will find is that they will spoil and get moldy faster because you don't have the sugar in there to throw off the osmoregulation of the microbes that want to grow in it. So water balance is a little bit different in cells with walls. Turgor is the firmness due to the cell swelling and putting pressure against the inelastic wall. That's going to put pressure back on it to oppose further water intake, but it's a healthy state for most plant cells. So here, in this hypotonic solution, you've got water coming into the cell and it's pushing hard against the cell wall. That's going to help give it its rigidity. In this next one here, when it's in an isotonic solution, it's going to become flaccid. And then here, when you put it in a hypertonic solution, it can become plasmolysed, where the cell immersed in a hypertonic solution will lose water and shrink. This will cause the plant to wilt and eventually kill it. So with facilitated diffusion, we have passive transport, but it's going to be aided by proteins. So diffusion and passive tell you it's not using any energy. However, it's facilitated. It's got a specific protein where this is going to happen. So certain hydrophilic solutes will cross passively with the help of the transport protein. Most of these transport proteins are going to be pretty picky about who they're going to help. They're going to be very specific, and they won't help just any molecule that comes across. So water is small enough to cross the lipid bilayer, but it does it very slowly. So the aquaporins are going to facilitate this happening much faster. So they will allow water to cross the membrane. So here, this one is showing an example of a carrier protein where you're going to have the substance come in, it will bind to it, and the carrier proteins will change shape to move the solute to the other side. So with gated channels, you have a channel protein, and these are going to be ion channels that will open and close in response to stimulus. So the sodium channels in muscle are an example. So when there is the stimulus, it's going to cause the channel to open and it will allow things to cross. And then when the stimulus is gone, it will close. The glucose transporters, these will undergo subtle changes in shape, and this will translocate the solute binding site across the membrane. So active transport, this is when you're going to use energy to move the solutes against their gradient. So when you're moving things with their gradient, that's what wants to happen naturally. It's going to require energy when you're going against what wants to happen naturally. So these will need energy enabled to do this. This enables the cell to maintain internal concentrations of small molecules that are different from their environment, and they're going to get the energy from ATP. So our classic example of this is the sodium-potassium pump. This is about a third of your resting energy expenditure during the day, keeping these sodium-potassium pumps going. So what's going to happen here is it's going to exchange sodium for potassium across the membrane. So here, the cytoplasmic sodium is going to bind the pump. And when it does, it increases its affinity for sodium. And the sodium binding stimulates phosphorylation by ATP. So ATP will transfer this phosphate on here. The phosphorylation is going to cause this protein to change shape. So now you can see it's a different shape, and it's going to release the 3 sodiums to the outside. This new shape has an affinity for potassium. So the potassiums are going to come on and bind, and then when you release the phosphate, it will return back to its original shape and release the potassiums inside. So this will allow you to release a greater amount of potassium inside again, and then it will return to having an affinity for the sodium. So anything that uses that much of your energy is going to be pretty important in keeping the cells alive. So these ion pumps can actually help to maintain membrane potential. Membrane potential is the voltage across a membrane. So it's the electrical potential energy from having the charges separated where the cytoplasm is relatively negative. So just like a battery, you have positive and negative ends separated. You have positive and negative charges separated across the membrane. This will create an electrochemical gradient. It's a force that drives the cations into the cell and forces the anions out due to the concentration gradient and electrical force. So an electrogenic pump is one that maintains one of the main ones in animal cells that is going to move sodium and potassium, and it will generate the voltage across the membrane. Another type we have is a proton pump. This is one of the main electrogenic pumps of plants, fungi, and bacteria, but we also use it in our mitochondria for ATP synthesis. It's actively going to transport hydrogen ions out of the membrane. So this can be used for ATP synthesis. What's going to happen is you will allow these to come back, but in response for allowing them to come back, the cell will require them to put a phosphate on ADP and make ATP again. So cotransport, this is coupled transport by a membrane protein. So an ATP-powered pump that's going to transport a specific solute is going to indirectly drive active transport of other solutes. So the substance gets pumped back across the membrane, and it will do work as it moves back. So this is how plants are going to translocate sucrose against the concentration gradient with the hydrogen ion. So here you can see you've got this proton pump. It's going to pump the hydrogen ions across the membrane. They want to come back to this side where the negative charges are. So the sucrose cotransporter will allow them to come back, but in exchange it says you're going to bring back a sucrose at the same time. So this is how we're going to move smaller things across the membrane. Sometimes we have to move things that are a little bit larger, and that's where bulk transport across the plasma membrane is going to come in. And we use exocytosis and endocytosis. Exocytosis is going to be when the cell excretes a biological molecule. The biological molecules are larger, and it will do this by fusing a vesicle with the plasma membrane. Once it does this, it spits out its contents. So the way I remember that this is removing something is I think of exo and exit. A lot of the secretory cells are going to use this. The beta cells making insulin, your neurons making their neurotransmitters. So endocytosis is going to be the exact opposite. This is going to be when the cell takes in a biological molecule. It may be in particulate matter and form a new vesicle from the plasma membrane. So this is the opposite of what happened with endocytosis or exocytosis. So with endocytosis, we have 3 variations of it. Phagocytosis, the term means cell eating. The cell is going to engulf a particle by wrapping pseudopodia around it. So here is your particle, and you can see the pseudopodium here that's wrapping around to engulf the cell. The particle is then going to come in, and it's going to be digested after this vacuole fuses with the lysosome. With penocytosis, this term means cell drinking. Here you've got droplets that are going to be taken into the vesicle, and then those will be taken in. The solutes that are in those droplets would be taken in as well. With receptor-mediated endocytosis, this allows you to acquire bulk amounts of substances. So even though they may not be concentrated in the extracellular fluid out here, what you have are these little receptor proteins in these coated pits that are going to wait for them to all be full. Once they are full with that specific substance, it's going to form a vesicle. So we refer to these as ligands that are going to come in. So the coated pit will form the vesicle containing those ligands, and then after it's ingested, the material from the vesicles and receptors are recycled.